This is quiz number 2. It's on the microscope, the parts of the cell, and cell division. So question 1 and 2, name the parts on the microscope, parts that are numbered. Number 1 is the ocular or eyepiece. And number 2 is the coarse adjustment knob. Three and 4, again, name the parts on the microscope. Parts that are numbered, 3 is the arm, and 4 is the stage. Number 5, the objective you should start with when focusing a slide is always the 4x objective. This is also called the scanning objective. Number 6, calculate the total magnification of the slide here. The slide here is focused in on the 40x objective, and the ocular that we use is always a 10x ocular. So 40 times 10, that means the total magnification is 400 for this slide. Number 7, name the 40x objective. It's called the high and dry objective. 8 and 9 were parts of the cell that's at the pointer on the microscope slides. I don't know if I can get this to actually focus. I probably cannot, but I will try my best, okay? You can see the flagella of the sperm. So flagella is the answer for number 8. And number 9, these are red blood cells. The pointer is on the plasma membrane. Number 10, the part of the cell at the pointer on the model is the mitochondria. Again, the parts of the cell on the model here. Number 11 is the nucleolus, and number 12 is the Golgi. You could say Golgi or Golgi body. Golgi complex would also be acceptable. And number 13, name the numbered part on the cell. The other instructor needed the model, so we have moved it, but 13 is on this organelle here. And this was the smooth ER, smooth endoplasmic reticulum. 14, name the process involving the diffusion of water molecules. Notice I underlined water. Diffusion of water is called osmosis. 15, experiment like we had done in the laboratory. What type of tonicity is the beaker solution? Okay, in the cell there's a 90% water environment, since it is 10% salt. In the beaker, there is a 60% water environment. Remember, water moves from high to low. So water is going to leave the cell. The cell will shrink. That means the beaker is a hypertonic solution. Hypertonic should be the answer. will the cells stay the same size, swell, or shrink? And this is the same example as the one that I just went over. And again, this is 90% water. In the cell, 60% water outside. Water moves from high to low, so that cell will shrink. Shrink is the correct answer. What molecules were able to diffuse into the cell? Well, this was the experiment we did in which the starch molecules were so large they couldn't pass. They got stuck in the cell. But the iodine molecules, being very small, were able to move, diffuse through the cell membrane from high concentration to low. So the molecules that are able to diffuse into the cell, you would tell me IKI or iodine. And you can also tell me water molecules were able to diffuse. The iodine is going to move from high concentration of iodine to low. Starch molecules could not move because they're so big. The water molecules are also going to move from high concentration to low concentration. So IKI or iodine and water, the correct answers for this. Was the starch able to diffuse out of the cell as we just went over? This is the same example. No, the starch was not able to diffuse out of the cell because of its size. Name the phase of mitosis shown here. This is telophase. The phase of mitosis shown here, anaphase. This is prophase. Structure at the pointer is the centriole. I will take centriole or centrosome. (23), the structure on the model at the pointer. I'm pointing to the nuclear envelope. Name the phase of mitosis at the pointer under the microscope. If I can have some luck like I had before. Maybe or maybe not. Usually very good with this, so I apologize if I'm making you all dizzy. Kind of. It is metaphase at the pointer. I think you can kind of see it. There we go. Metaphase. That is so hard to do, just so you know. Students do it much better than I do. Phase at the pointer again under this microscope. And this was prophase. There we go. Let's see here. Prophase at the pointer there. Chromosomes are scattered, and you can tell that there is a nuclear envelope still around those chromosomes. So prophase is the correct answer for 25. And that's the end of the review for quiz number 2. I hope everyone did well. Certainly see me before or after class if you have any questions.